Hello, everyone. I got a request from my Med 1 friend to do the GNH, Genetics and Health Week 3 GLS. And so I'm going to be doing activity 2, some of the Mendelian genetic problems to tackle, and doing a walkthrough of my own thought process, and hopefully it can help other students this year and into the future as well. And it also links to biology as well. I had a look at some of the biology externals. I didn't do biology in year 12, but I had a look, and some of this content actually covers through, so it can also help if any of you biology students are interested in watching me do this. So let's go straight into it. Question 1A. We just have to predict the phenotypic ratios of the offspring. Okay, because we have a dominant A and a recessive A. So we know we're just dealing with normal Mendelian genetics. Okay, so for A, part A, we'll just be doing a Punnett square. So if we just write out our individual genotypes for each parent and then doing a table like this, where we have 4 boxes to cross over between them, then we know that our, if we just do from left side to the top, we have the following in green, okay? And because the capital A is dominant, then that means that um, the phenotypes of capital A and lowercase a are just going to be the dominant phenotype. And the double recessive alleles are going to be the recessive phenotype. Noting that we need 2 recessive alleles in order to display the recessive phenotype for a Mendelian genetic problem. So in this case, we have 2 heterozygous and 2 homozygous recessive, and that just makes a ratio of 1 to 1. Okay, so that's our answer there. And I'll let you do B and C on your own. For incomplete dominance, it's a bit different. All right, so if we were to do, say, um, option A again, it'll be the same thing. So I'll just copy this just to save time. Okay. What's the difference here is that when we have a heterozygous, it's actually called something called the intermediate phenotype. Okay, so say we have A, capital, the dominant homozygous is red, flowers per se, and then we have the recessive as the white flowers for homozygous. Then a heterozygous will actually not be red because although it is dominant, incomplete dominance means that we have the blending of 2 traits. So if we were to do an average of red plus white, then we would get pink, okay, for example. So heterozygous alleles will actually show an intermediate phenotype, something in between. That's what the intermediate means. Okay, and so for this one, we actually have it still be a 1-to-1 ratio, but we have to be specific in saying 1 intermediate to 1 recessive. Okay, and I should actually be more specific on question 1 here. So I should say 1 dominant to one recessive. Okay. Because one-to-one ratios, if we just write the numbers, they don't really mean anything because they can, um, you might lose marks if you don't necessarily be specific on what each ratio or each part in the ratio actually means. Okay, just keep that in mind. Okay, and then with codominant, so if I just do C, part C, for example, I'll do, these are all capitals here. All right, then I just draw in a different color the phenotypes of the children. Okay. Sorry, that's going to be both capital A. My bad. Then we can see here that we have one homozygous dominant, A, and then two of them are the heterozygous AB, and then one of them is the homozygous capital B. Okay, and what this basically means is that we have one of them will just be, say, have the A phenotype, and then one of them will have the completely B phenotype. But instead of the blending together, what codominance means is that they will coexist together at the same time. So this is basically both A and B. So as opposed to incomplete dominance where there's a blending, codominant actually means that the AB phenotype um, will actually show both A and B phenotypes completely, but separately. Okay. So for example, if our red and white flowers were codominant, you'd get something like this, right? So you'll have a bit of red, okay, in the flower, and this is complete red, okay, complete red, but then maybe the other half will be pink. Okay? So essentially, rather than them blending together to make an intermediate phenotype, we have both of the phenotypes, red and pink, expressing, okay, in the flower. That's what codominant is, and that's what represents by the genotype capital A, capital B. All right. So to express the phenotypic ratios, that would just be one dominant dot dot two intermediate dot dot one recessive. Okay, to represent the um, uh, homozygous A, heterozygous AB, and then homozygous BB, assuming that, of course, wait, sorry, it's not actually talking about recessive here. We're talking about one AA, two AB, and one BB. We can't say that they're dominant or we can't distinguish whether they're recessive or not because they're co-dominant, both of them being dominant really. So um, we can't really say which one's a lowercase or recessive and stuff like that. Okay. Moving on. Question 4. In humans, brown eye B are fully dominant over blue eyes. You visit two families, the Joneses and the Irvings, when the parents are out of town. The children of Mr. and Mrs. Jones show an approximately 3-to-1 phenotypic ratio of brown eyes to blue. What are the genotypes of Mr. and Mrs. Jones? Okay, now if we were to um, show a 3-to-1 phenotypic ratio of brown eyes to blue, then we know this is a typical phenotypic ratio we have in two heterozygous genotypes. Okay, because we know that one out of four possible combinations, which is the total of this ratio, are possibilities, we'll say. One will be recessive. Okay, and the only way to have that is if we have the recessive gene, obviously. Um, that are possible from the parents, right? So, but the fact that there's a 3-to-1 ratio, it means that the dominant phenotype dominates, which indicates to us that we need to have a heterozygote. Um, both of them need to be heterozygote. Okay, so if we just do that here, and we can see that we have three of them as dominant. Okay, because just the presence of a dominant allele will make them the dominant phenotype, but two recessive alleles will make it a recessive. Okay, and that gives us the 3-to-1 phenotypic ratio. So therefore, these, as I circled in black circle there, are the genotypes of the parents. So we would just say heterozygous for both of them. Okay. However, if there's an approximate 1-to-1 phenotypic ratio, then that means we have to have 50% dominant, 50% recessive. Okay, so we need to have the presence of a dominant and a recessive, but they need to be, I guess, like 50-50. So the way we can do that is just to think about it's like a puzzle, really. I think that's the best way to go about it. If we have the heterozygote, so we can both have the recessive allele as well. And we make one of them be recessive, then maybe that'll increase our chances of having the recessive allele for the offspring, and therefore we'll be able to increase that chance to 50% rather than dominant phenotype dominating with a 3-to-1 ratio. So we'll give that a go. So therefore, their phenotypic ratios of the parents are B, capital B and lowercase b, by lowercase b, lowercase b. Okay. Question 5. Provide a brief outline of Mendel's law of segregation. How does meiosis explain Mendel's law of segregation? Okay. So for this one, basically, I'll just write down Mendel's law of segregation, but it just basically means that the alleles, um, the allele pairs segregate. So each gamete carries one allele homologous um, chromosomes segregate in meiosis one, explaining the 1-to-1 gametic allele ratio. Okay, so basically what we're talking about here is that members of a gene pair separate from each other in gamete formation. Okay, so if we have a heterozygous, then a genotype from an individual, then the A would go into one progeny cell for the capital A, and then the lowercase a will go into the other progeny cell after meiosis 1. So they basically segregated from each other. Okay? And it basically derives from that fact that during meiosis, alleles, which is one from our biological mother and one from our biological father, separate into different gametes. Okay? So, question 6. Seed color in pea plants is controlled by a single gene. Consider the cross of a pure breeding yellow seed color variety to pure breeding green seed color variety, in which yellow seed color is completely dominant. Okay, so let's just say that let capital Y equal the um, yellow seed dominant. Okay, that means lowercase y will be the green seed recessive. Okay, so if we have the pure breeding yellow seed, that means that we have a homozygous dominant, and a pure breeding green seed will be a recessive homozygous. Okay, so making our Punnett square here, we just know that every single one of these will have a dominant allele and a recessive allele, so all of them are going to be heterozygous. But in terms of the um, phenotypes of the F1 generation, right? We know that if there's 175 F1 plants, then that means that out of these combinations, these 4 possible combinations, 175, so all will be yellow because this is because yellow is the dominant trait and there is a capital Y present in all of these possible combinations, and that leaves with 0 green. Okay, so yeah, essentially that's what we've done in B. All right, phenotypes are sorry, the parents are homozygous dominant and homozygous recessive, and then the first generation, familial generation 1, will have all heterozygous genotypes. All right, but if we have the F1 plant self-crossing, then what will happen is that we have the Punnett square of 2 heterozygous. If we make that, then what will we get is this 1 to 2 to 1 ratio for the genotypes, as we have learned, right? So we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay, but the phenotypic ratio will be just combining these first two genotypes to make a 3-to-1 ratio, for 3 being the dominant trait, which is yellow, and 1 being green. Okay, so just applying basic maths, my ratio 3-to-1 has a total parts of 4, so 3 plus 1, and then that means out of 400 resulting familiar generation 2 plants, each part will equal 400 out of 4, which is 100. Okay, and then putting that into the ratio here, we have 3 times 100 yellow, which is 300 yellow, and 1 times 100 green, which is 100 green. Okay, and this is out of 400, right? That answers that question there in terms of how many should have yellow and green seeds. Okay, and as I said, the genotypic ratio is what I've just mentioned there. Okay. Now, providing the possible genotypes for the blood types listed below. Okay, so we know in terms of the ABO blood type, we just have a capital I for being dominant or co-dominant, right? So type A will either have the homozygous A, or because type A is dominant, we can have a lowercase I and a capital I raised to the power of A. Okay, so these will both manifest as a type A phenotype. Okay, both of these genotypes. Same thing with B, right? It's exactly the same, just with B instead of A. Okay, so those are the two genotypes, but there's only one genotype for type O, okay? So the type O genotype must be recessive, homozygous for the lowercase I, okay? And then with type AB, we know that there's only one combination where we have two dominant alleles, but they're coexisting, so they're co-dominant. Okay, so that's capital I to the power of A and capital I to the power of B, but all uppercase there. Okay. Now for this one, a man with blood type A and a woman with blood type B have 3 children, a son with type AB blood and 2 daughters, one with type O and one with type B blood. What are the genotypes of the parents? Okay, now the key point there is that we have a daughter with the type O, okay? And we know that type O is like a recessive homozygous, okay? Which is the lowercase I, 2 of those, right? Now, the only possibility for that to happen, if the father, has blood type A and the woman has blood type B, the only way they can pass down a recessive lowercase I is if they are actually, they have that allele. So as we can see here from question 7, so I've underlined here, they must be carrying that lowercase I allele. Okay, so therefore, the father's genotype is capital I to the power of capital A, lowercase I, and then the mother's is capital I to the power of capital B, lowercase I. Okay. Because in that case, if we just quickly draw a Punnett square to check, we know that it is possible, since they carry that, sorry, this one should be just lowercase I, since they're carrying the recessive lowercase I, then 25% of the time they will have a child with recessive lowercase I. Okay, and that's the possibility there. Okay, Whereas, obviously, if one of them or both of them didn't have the recessive um, lowercase I, then that means that um, you can't manifest a type O blood type because you need to have two, one from each parent, two lowercase I alleles to manifest an offspring with a type O blood, which is um, having a genotype of what is circled in pink there, right? Question 9. What is the chance that a woman with type AB blood and a type man with type A blood, whose father is type O blood, can produce a child that has? Okay, so in this case, we have some key information. So we know the woman has type AB blood, so we can immediately definitively say that one of the parents is um, type this genotype. All right. So hopefully you understand that now. I won't be repeating myself with the genotypes, especially with blood group can be a bit um, what's to say. So we have the man with type A blood. Okay, so we could either have homozygous A or with the lowercase I, and we need to choose which one is more likely or which one is the correct one. Okay, so if the father is type O blood, then that means the father's genotype is lowercase I. Okay, so that means no matter which allele they choose, whether it's the first lowercase I or the second lowercase I, their offspring are always going to have that lowercase I. So it has to be this genotype I've circled in black there. Okay? So therefore, this is the Punnett square that we're meant to be using. Okay. So I'm just going to be doing that. Okay, and from here on, this is just simple maths now. Okay, we have 4 combinations, and we're trying to figure out which one have type A blood. Well, that's going to be this one and this one. First two on that first row right here. Okay, so that's going to be 50% because that's 2 out of the 4 possible combinations. Type B is going to be this one, right? So that's 25%. Type O blood, 0%. We don't have a homozygous lowercase I. And type AB blood, that's this one right here. So that's going to be 25%. It's 1/4 of the possible combinations. Moving on. An unnamed human trait is controlled by a single gene. There are 4 possible alleles for this gene, W, X, Y, and Z. Alleles W, X, and Y are all codominant, whereas the Z allele is recessive to all the other alleles. Okay, so how many phenotypes are possible? Okay, so this takes a little bit of problem solving, but if we just give ourselves some space here and we think about the different letters here and we try and do a table, sort of like a Punnett square. Just bear with me here. And we can figure out the phenotypes and the genotypes, okay? So I'll be going from left to top, okay? Feel free to fast forward if you wish. Just being very careful here. I'm being consistent from left side to the top in that order. Okay. Right. So we know that W, X, and Y are all codominant. So let's think of, say, phenotypes with um, that we can find from here. So um, we can see that we have four phenotypes along this diagonal here. Okay, so that's going to be our phenotypes for simply capital, sorry, um, for our, say we have just a double up homozygous for each letter, so that's going to give us W, X, Y, and Z. Okay. However, we know that since Z is the um, recessive allele, it's not codominant like the other three, then everything on the Z line will be included in these phenotypes. Okay. For example, ZW, that's going to express as a phenotype of just W because W is dominant. We assume that because it's codominant with other alleles, and so it will, um, I guess, manifest over the recessive Z allele. Okay. So same with ZX, ZY, and you can obviously see how like WZ and XZ are going to be just the same thing, right? They just swapped around letters, but it doesn't matter the order of the allele combinations as long as they're present and we know how they relate to each other. Okay. Now, time to look at the codominant phenotypes. So we know that if we have WX is one of them, all right. Same as WY, those are two WYs, and then YX are those there. Now I'm just making sure I've ticked everything. Oh, I forgot a XW there. So it seems like every single, if I just color this in with a different color, we have three extra codominant phenotypes where these are all codominant X, Y, W, X, and Y. All right, so we have the XW phenotype. I'll write it in the answer area. So we have the um, WX phenotype, the WY phenotype, and the XY phenotype. And so if we count up those phenotypes, then those are 7 phenotypes. Okay. Question 10b. How many genotypes are possible? So this is essentially trying to look and see, okay, I'm not talking about the phenotypes or what traits they present are expressed as, but just the individual letters or alleles combinations that we have. So we know that we can just use the same technique as we did for phenotypes. We just take this diagonal from top left to bottom right to get the homozygous allele combinations, okay? Because we know those are going to have their own genotypes. So we have lowercase w, sorry, doesn't matter to be lowercase, but I'm just giving the example. The case standard doesn't really matter here in this case. I'm sorry, let me do it. So we have those 4 genotypes, and then we're having a look now to see what are the other combinations. So we know XW and WX are just the same genotype. They're just swapped around letters, but it's still the same allele combination, okay? Because order doesn't matter here. And so that's would be an extra genotype. We have YW as well, so that's another extra genotype. Okay, so just going down on the diagonals, as you can see, just essentially this way, and basically looking at what hasn't been crossed yet. So in this case, we have YX and XY. Okay, so you can see the sort of symmetry here from the diagonal, and this is how I like to go about my questions because they can help um, with making sure you don't miss anything as well. Okay, and then same with zx and zx, you can see that symmetry there, and then last but not least, we have zy and yz, which are the same there, okay? And so if we have 4 plus different colors we have one yellow, green, blue, purple, and stuff like that. So we have 1, 2, 3, 4, 5, 6. So we have 4 plus 6 equal 10 genotypes that are possible, okay? And if we want, we can be specific and write those down. So we have X, Y, W, Y, W, X, Y, Z, W, Z, X, and Y, Z, okay? If we wanted to be more accurate, we might put them in alphabetical order, but it doesn't really matter. As long as you're answering the question, we have 10 possible genotypes. Okay. Snapdragon flowers are normally white or red. This is question 11. A botanist observes a strange variant that has pink flowers. Okay, so we know immediately we're talking about incomplete dominance. Okay, for heterozygote alleles, right? So if the pink flower variant, so the two heterozygotes are self-crossed, that's what that means by two heterozygotes, and the following phenotypes are observed, which we have, as we can tell here, just from a wide scope perspective, that we have that 1 to 2 to 1 ratio. Okay, we have, so say one part is just 30, two parts would make it around 60, and then one part would also be that 30 around that number. Okay, so the reason why it's not exactly the same is because this is experimental observed data, not the expected or theoretical value for that 1 to 2 to 1 ratio. Okay, there'll always be a bit of slight changes, okay, and a variability in the real world, okay? But what we can know here is that we have we can definitively know that the genotypes of the parental flowers has to be heterozygous in order to show this 1 to 2 to 1 genotypic ratio, okay? So those are the genotypes of the parental flowers. Let's write that properly there. And the genotype of the offspring with red flowers are therefore going to be, we draw a Punnett square, okay? We know that we have a 1 to 2 to 1 ratio where we have the genotypes as follows. And the ones with the red flowers are going to be the capital A genotypes, okay? Because we know that red flowers is going to be dominant over the pink flowers. All right. I think that's just assumed knowledge from this question. But yeah, I guess it's just a typical co-dominant example that you just have to be aware of in terms of snapdragon flowers. Okay. Question 12. In East Asia populations, two types of earwax are observed, dry and sticky, which is also smellier. A geneticist studied the inheritance of this trait through observing the children of parents with different combinations of earwax phenotype. The results were as follows in this table, okay? Based on this data, what can you conclude about the inheritance pattern of earwax type? Okay, so as we can see in the data, we have the sticky earwax phenotype of the parents. Once those are present, we know that we have most of them are going to be sticky in their offspring, okay, for the earwax. And so, however, we know that when two dry phenotypic earwax parents have offspring, then we're not going to get any sticky phenotypes from the children or the offspring. They're all going to be dry phenotypes. Okay, so what can explain that? Well, the way we can explain that is the fact that earwax seems to be inherited as a simple autosomal dominant or recessive Mendelian trait. Okay, where obviously we would have the sticky as dominant and the dry as recessive. And we know that sticky is dominant and dry is recessive because the crosses between dry individuals would produce only dry offspring. Okay, and the reason for that is because if dry was recessive, that means we would need to have two recessive alleles in order to express that phenotype. Okay, so say our recessive allele was lowercase a. Okay, if we crossed two recessive alleles, then we know that all of their offspring are going to have dry earwax. Okay, right, there is no possibility for that dominant phenotype, dominant genotype, which is the capital A in this case, to express itself in the genotype and therefore to be expressed in the phenotype. Okay, the actual earwax of the offspring. Okay, and so that's what's happening in this bottom row here. We cross two dry phenotypic parents. All of them, all of their children are going to have dry earwax. Okay, none of them, or zero, as shown here, will have sticky earwax in the phenotype of the children. Okay. However, if we have the phenotype of parents with sticky earwax, whether they're both of them are sticky, have sticky earwax, or one of them only has sticky earwax, then they would give rise to both types, okay, sticky and dry, because in this first row here, we could have a possibility of heterozygote parents crossing over, and that will result in um, some children having the dry phenotype, where we can only where the alleles that get passed on are both recessive phenotypes from each parent. Okay, and that would result in dry earwax. Okay, so that's a possibility. Although both of them are sticky, uh, both of them have sticky earwax, the parents, then we can still have that possibility of having the dry and the sticky phenotypic children. All right, so that's what is evident there. It's not necessarily the numbers, as I'll get to part B. It's not telling about the ratios of them. That's not really what's relevant. What's relevant is that a phenotype can only be expressed in a recessive allele if both of them, if both of the recessive alleles are present in the genotype of that individual. Okay, so we cannot determine from a phenotypic sticky earwax parent or individual or anyone, we cannot determine whether they are homozygous dominant or heterozygous. Okay, we don't know, but what matters is that there's a possibility of that happening, and therefore we can determine which one's going to be a dominant, which one's going to be a recessive phenotype. All right. So moving on to part B, then we are basically trying to ask, why are there no 3-to-1 or 1-to-1 ratios in the data? Well, because in order to get Mendelian ratios, we need to examine pure breeding individuals with known genotypes. Okay. However, we had unknown combinations of homozygous capital A, and heterozygous lowercase a, capital A individuals with sticky earwax. Okay. And in terms of the pure breeding, only the dry lowercase a individuals can be expected to be pure breeding. Okay? Because as we remember before from the previous questions, Mendelian's laws of the 3-to-1 and the 1-to-1 ratios are a result of pure breeding um, parental um, generations, right? So if we have this happening, we know that they're homozygous, then we know we're going to get the heterozygous for all of the offspring. And if those self-breed, because we know the specific genotypes, not just their phenotypes, then we know we're getting that 3-to-1 ratio for the offspring, okay, for that 3-to-1 ratio. But because we don't know the actual genotype of the parents, we can't definitively have a consistent 3-to-1 ratio, okay? This sticky, again, can either mean homozygous dominant for the sticky allele or heterozygous for those alleles. Question 13. In humans, hitchhiker's thumb is a dominant characteristic controlled by a single gene. Part A says a man who does not have hitchhiker's thumb has children with a woman with hitchhiker's thumb whose mother lacked hitchhiker's thumb. What proportion of their children would you expect to have hitchhiker's thumb? Okay, so I've made some previous notes before, but essentially because we know that hitchhiker's thumb is dominant, then we know that if we let H be our letter here for the alleles, then a person who does not have hitchhiker's thumb will have the recessive homozygous allele genotype. So we know that this is the genotype of the man, okay? And then we are crossbreeding with a woman with hitchhiker's thumb whose mother lacked hitchhiker's thumb, okay? So that means the mother of that woman, or the mother in this case for these children, so essentially the grandmother, lacked hitchhiker's thumb, so that means their genotype was also a lowercase h, which means the only allele that they could give to offspring, which would be this woman right here, has to be a lowercase h. So that means that the woman has to be heterozygous for the dominant characteristic, okay? Hitchhiker's thumb, right? So that is the woman's genotype, okay? We know that because they must have gotten the recessive lowercase h from their mother, because that was the only, those were the only alleles that the mother could give for this woman, okay? But now that we have this new combination for breeding, then the question asks us, what proportion of their children would you expect to have hitchhiker's thumb? So If we do a Punnett square, to find our possible outcomes, we know that 50% are going to have the dominant allele, which is the capital H, and therefore have the hitchhiker's thumb. Okay, because that dominant capital H allele is going to express in the phenotype, right? Part B, a man with hitchhiker's thumb and a woman who lacks hitchhiker's thumb produce a child who lacks hitchhiker's thumb. Okay, so essentially what it's saying is that um, a man with hitchhiker's thumb, so they must have at least one allele for the dominant allele, crossbreeding with a woman with hitchhiker's thumb, sorry, who lacks hitchhiker's thumb, so they must be homozygous recessive, will produce a child who lacks hitchhiker's thumb, so also homozygous recessive. So the only way that can happen is if the man has a genotype that has a recessive lowercase h to be passed on to that child. Okay, so in that case, they can pass on this lowercase h, and the mother can only give lowercase h. So it doesn't matter which one gets passed on, it will transfer to the genotype of the child, and that manifests as lacking hitchhiker's thumb. Okay. In part C, a man with hitchhiker's thumb and a woman without hitchhiker's thumb produce 8 children, all having hitchhiker's thumb. Can you be certain of the man's genotype? Why or why not? Okay. In this case, no, we cannot. All right. The man's genotype could either be H capital, both of them, or capital H and lowercase h, right? So, for example, if the man was heterozygous, right? And so we have that same pedigree as we had before, where we have that 1-to-1 ratio or 50%. Then, so as we said, there's a half chance that each child they have as offspring will have hitchhiker's thumb. Okay, if 8 of the children, all 8 of them will have hitchhiker's thumb, that's the probability of 1/256. Okay, but that is still possible. Although very unlikely, it doesn't definitively tell us whether, if we know that 8 children, all of them having hitchhiker's thumb, that doesn't definitively say whether the man's genotype is homozygous dominant, even though it's more likely to be that. It doesn't, with certainty, 100% certainty, say that the man's genotype is a specific homozygous dominant, because we know that there's still this chance, this 1/256 chance, that it can happen if the man is heterozygous. Okay. Question 14. So we have 2 pure breeding lines of plants, one with white flowers and one with red flowers. The resulting F1 generation all has pink flowers. Okay, and then a subsequent self-cross of the F1 generation results in the following numbers for these offspring. Okay. And then here we're trying to ask if Incomplete dominance may explain the inheritance pattern observed. Okay. And you have to decide to perform, you decided to perform a chi-square test to see if there is statistical support for an inheritance pattern of incomplete dominance. Okay, and I have the pages for these notes in my OneNote just over here. So I'll be referring back to those later on. Okay. All right. So if incomplete dominance is responsible for the observed inheritance pattern above, what numbers of plants with each flower would you approximately expect to see in the F2 generation? Okay, so note that this is our observed values, all right? But now we're trying to find the experimental or expected values. Okay, keyword there is expect. Okay, so we'll basically just repeat this table up top again, but with different numbers. So we have pink flowers, red flowers, and white flowers. Okay. And we know that if all of the F1 generation is pink, that means they're heterozygous. So let's say that um, we're using A as our letter. So crossbreeding two heterozygotes because we're crossbreeding in for the F2 generation is a self-cross of the F1 generation where they're all pink. So that will give us that 1 to 2 to 1 ratio where half of them are going to be the pink flowers, which is, or in other words, 2 over 4 out of the 4 parts here. And then 1/4 will be completely homozygous for red, and 1/4 will be completely homozygous for white. Okay, so we know that's true because it does add up all to a value of 1, these proportions. But in a um, total of 320, which is what we were given in the question, then we can use that to make our expected values. So if we have 4 parts in the ratio 1 to 2 to 1, then 1 part is equal to 320, the total divided by 4, which is 80. Okay, so we can know that about half of the total, or like 2 parts of um, 2 parts, so 80 times 2, 160, will be the intermediate pink flowers, and that's what that 2 is representing here. Half of them, 1/4 of them will be 80 and 80. Okay, so this is of the total. Now we have to use the chi-square formula to calculate the chi-square value for the above scenario, all right? So we can do a table up where we have the um, observed value and the expected value. Okay, so you can see there's lots of tables in G&H, but we'll work with them. Okay, so we know that the observed values we just take from the table. Pink flowers are 182, red 161, 77. Expected values are what we calculated in part A. Okay, and then now we just do a chi-square test where we the formula which we have to memorize is the so this is chi-squared, that's what the symbol for it. It's the sum of the square of the difference between the observed and expected values, so the squares of those over the expected value. Okay, so essentially what's that saying is that we need to calculate every value for this fraction per phenotypic category, all right, and then add them up. That's what this sum means. Okay, so um, what we can do now is we can separate them into labels, say like pink, red, and white, and then just add them together, right? So for pink, let's just focus on this line right now. So this first row. So we have observed value of 182 minus 160. Square that and then divide by the expected value, which is 160. And then plus my value for red. Now looking at this row, so I have 61 minus 80. Don't forget the squared. And the reason we squared that is because if you notice, if we just did 61 minus 80, that'll give us negative 19. And we don't really usually want to work with negatives in our answer because it will just, um, it's not significant to have a negative and it doesn't help really with the calculation we're trying to find for the chi-squared test. Okay, so just remember that squared all the time. It'll be over the expected value just on its own. Okay, and then adding the white row. So again, it's you just gotta practice these sort of questions, chi-squared tests, and um, I know in week 8 this week we'll be doing chi-square as well for population genetics, but it's really just the same pattern happening. And if you use your tables and everything, then it'll get really easy to solve these problems, and you'll be experts at it very quickly. It just takes a bit of practice, but there is a clear pattern here, all right? So another way to check your work, it's like a dummy check, is making sure these values up here and on the bottom are the same, okay, which in this case it is. All right, and then we can just plug this in our calculator, and then we get a value of 7.65, okay? So it's really crucial you have those 3 lines of working out. You have the formula, which is tick, could be half a mark, and then substitution, okay, tick, which is could be another half a mark on the exam, and then another tick for this one for the answer, the proper accurate chi-square test. Say we round it to 2 decimal places. Yep, so that's the calculate part. Okay, you're not going straight to the calculate section because you can lose 1 whole mark from that. Okay, how many degrees of freedom does this scenario correspond to? So we have a formula, it's very easy to remember. Basically, the degrees of freedom equal number of phenotypic classes or categories minus I won't put a slash there because it might think like might seem like a fraction, but it's not. Minus 1. Okay, so basically it's trying to ask us what are the number of phenotypic classes or categories. So what are different type what are the number, sorry, how many different phenotypes are there in this scenario? So there's 3, okay? You can have pink, red, or white. 3 phenotypes. And then take 1 from that. So our degrees of freedom is 2, okay? Essentially what degrees of freedom is saying is that once we know the number of degrees of freedom, so say for this example, if we know the values or like I guess proportions for 2 of the phenotypes, then we already know 1. Okay, the third one will be fixed, or the one left over, because we just basically do 1 minus those 2 proportions. Okay, so that's what it essentially means, degrees of freedom. But in terms of what we really need to know is that how to use the chi-square test to evaluate whether our hypothesis is um, is met here. All right. So I'm going to go to the document, and here are the probability values for the chi-square analysis. Okay. So what it's talking about here is we have the degrees of freedom on this column, this first column right here. All right. And we look for our degrees of freedom. So we know that was 2. Okay, 3 minus 1. And we look at this row now. We just focus on that row. Okay. And then we have a look at our number for our degrees of freedom on this row. Okay, because imagine this as like a range. Okay. So say this is like a range of values. Okay. So I know there's specific values put into this table, but that just indicates the probability um, that our hypothesis is true, for example, okay? But obviously, in terms of the chi-square test, that's really talking about difference, like from our observed and expected value from our prediction or hypothesis. So the greater the chi-square value, the more we're going to have variability, and it's going to contradict between the observed and expected results. So since we got a chi-square value of um, 7.65, we look at the table again and we have a look. Okay, so 7.65 are along here. Actually, that's going to be past my 0.05% probability that um, supports my hypothesis. Okay, so my 7 point, what was it, 65 is essentially outside this acceptable range for my chi-square values, which means that I mean on an the hypothesis I've stated saying that this is a incomplete dominance scenario is unacceptable. Okay, so I'll show my working out saying that at df equals 2, 7.65 is greater than 5.99, so I reject 1 to 2 to 1 at a probability of 0.05 and doesn't fit perfectly. Okay, so that's the 5% due to chance sort of thing.